Welcome back to our series on analyzing creationist claims. Today, we're going to take a look at radiocarbon dating. How does it work? How do we know it's accurate? And what about creationist claims that it just doesn't hold up when put to the test? Well, let's dive in. First off, what is radiocarbon dating? Well, simply put, radiocarbon dating is a method for determining the ages of things that have lived within the past 50,000 years or so. It's perfect for ancient wooden tools, or artifacts from early civilizations, or the collagen preserved in Neanderthal bones. For anything older than that, different radiometric methods are used. We can save those for a different video. Where does radioactive carbon-14 come from? Let me start by saying that carbon-14 and radioactive carbon-14, those are the same thing. So you may find me using those two terms interchangeably as we go along. The story of radiocarbon dating starts in the air around us. Air looks empty and it feels empty, but the air around us is actually made of molecules. About 78% of those molecules are nitrogen gas, or N2. That's two nitrogen atoms stuck together. Then about 21% are oxygen gas, a little 1% are argon, and about 0.04% of the molecules around us are carbon dioxide. And then there's a tiny pinch of other trace gases. Now, out of all of these gases, which one do you think matters most for radiocarbon dating? You might guess carbon dioxide, since that's where the carbon is. But the real starting point is nitrogen. As this N2 is floating around in the atmosphere and in the air around us, every now and then, some of those nitrogen molecules get impacted by high-energy neutrons. Where do those neutrons come from? Well, when stars explode way out in the galaxy, they send out shockwaves racing across space that carry high-energy leftover protons, traveling at incredible speeds. Some of those high-energy protons eventually reach Earth, where they collide with the molecules that make up our atmosphere. And it's those collisions that end up generating those fast-moving neutrons. These fast-moving neutrons can then go on to interact with nitrogen atoms and create carbon-14. How? Well, remember that nitrogen gas is N2. It's two nitrogen atoms bound together. Those atoms are what we call nitrogen-14, meaning that they each have 7 protons and 7 neutrons. If an extra neutron collides with one of those nitrogen-14 atoms, it can knock out one of those protons. With one less proton, that nitrogen atom is no longer nitrogen at all. It's been transformed into a carbon atom, specifically the isotope carbon-14, which has 6 protons and 8 neutrons. That carbon-14 atom doesn't stay by itself for long. It'll quickly bond with oxygen atoms in the atmosphere to form radioactive carbon dioxide. That's a CO2 molecule that contains a carbon-14 atom instead of the usual carbon-12 atom. From then on, those specially generated CO2 molecules behave chemically just like ordinary carbon dioxide, drifting and mixing in the air around us. A key point here. Almost all of the carbon dioxide in the air around us is made with carbon-12, but a very tiny fraction. Just 1 in every 1.3 trillion carbon dioxide molecules is radioactive carbon dioxide, containing carbon-14. A small trickle of new carbon-14 is created continuously in the atmosphere by that cascade of high-energy protons and high-energy neutrons colliding with atmospheric nitrogen. And it's those carbon-14 atoms that make radiocarbon dating possible. How does carbon-14 help us date old things? Plants, every plant on planet Earth, take in carbon dioxide from the atmosphere during photosynthesis. That's literally where the carbon in plants comes from. The carbon that makes up wood, stems, leaves, roots, all comes directly from the carbon dioxide in the air. Inside plant cells, The carbon atom then gets stripped off of CO2 and is used as raw material to build those plant tissues. Take a single oak leaf. It might contain something like 10 billion trillion carbon atoms. Almost every single one of those atoms is the stable isotope carbon-12. But thanks to that tiny fraction of radioactive CO2 in the atmosphere, about 20 billion of those atoms are carbon-14. That carbon-14 enters the plant along with the carbon-12 as carbon dioxide, and once it's there, it becomes part of the leaf's structure. But as soon as that oak leaf dies and falls off the tree, it stops taking in CO2. And from that moment, the amount of carbon-12 and the amount of carbon-14 that make up the leaf are fixed in place. You can think of it as locking in the ratio of carbon-14 to carbon-12. It's a ratio of about 1 to 1.3 trillion at the time of the leaf death. And here's where it gets interesting. Carbon-14 doesn't stay carbon-14 forever. It's unstable. Each carbon-14 atom will eventually decay back into nitrogen-14. A good analogy would be like ice melting into water. Left alone at the right temperature, ice will always melt into water. In the same way, C14 atoms will always decay into nitrogen-14 atoms at a steady, predictable pace. That pace is what we call a half-life. For carbon-14, the half-life is 5,730 years. That means that after 5,730 years, half of the carbon-14 atoms in a sample will have turned back into nitrogen-14. So if you started with 20 billion carbon-14 atoms in a leaf, after 5,730 years, only 10 billion will remain. After another 5,730 years, 5 billion remain. Then 2.5 billion, then 1.25 billion, and so on. The carbon-14 atoms steadily morph into nitrogen-14. Meanwhile, the carbon-12 atoms in the leaf stay constant. If at the time of death there were 10 billion trillion carbon-12 atoms, then after 5,730 years there would still be 10 billion trillion carbon-12 atoms. Then after another 5,730 years, there would still be 10 billion trillion carbon-12 atoms. And after another 5,730 years, there would still be 10 billion trillion carbon-12 atoms. You get the idea. The carbon-12 atoms don't decay or change into anything else. That contrast is what makes radiocarbon dating work. If we take an ancient leaf, or a piece of wood, or the collagen in fossilized bone, and measure the current ratio of C14 to C12, we can compare it to the known half-life curve of carbon-14, and calculate when that object stopped taking in carbon. In other words, when it died. This principle applies not just to plants, but to anything that depends on plants for carbon. Herbivores incorporate plant carbon into their bones, teeth, and tissues. Carnivores and omnivores incorporate the carbon from herbivores that they eat. Paper made from plants. Parchment made from animal skin. Wool, cotton, leather, wood. All of these things can be dated because they all ultimately trace their carbon source back to atmospheric carbon dioxide. What's the biggest creationist objection to radiocarbon dating? Perhaps the most common objection that you'll hear from creationist commentators is that this method of radiocarbon dating relies on shaky and unproven assumptions. And so the method itself just can't be trusted because of this. Let's hear them explain it.
So to carbon date something, you are assuming you know how much was in it when it started. It's assumed the ratio of carbon-14 to carbon-12 in the atmosphere would be the same found in plants and animals that are alive today. The atmosphere today is 0.0000765%, so they assume the plants and animals started with the same number. There's no way to know that.
You have to plug those numbers, those isotope ratios, into a formula, and the formula has assumptions like how much of each of these isotopes was there in the beginning when this creature died. No one knows, so you have to backfill that assumption with some fake number because you weren't there to measure it back then.
In essence, what they're trying to get across here is that maybe, just maybe, 10,000 years ago, the ratio of C14 to C12 in the atmosphere wasn't 1 in 1.3 trillion. Maybe it was 1 in a billion. Maybe it was 1 in 2 trillion. Who knows? We weren't there to measure it. So we have to either assume that the ratio in the past was the same as it is today, or we just make up some arbitrary ratio as a starting point. And since radiocarbon dating depends entirely on knowing the starting ratio, the method just can't be trusted. First, we can give them a tiny bit of credit here. They've actually keyed in on something important. The ratio of C14 to C12 in the atmosphere actually hasn't been constant over the last 50,000 years. It's fluctuated. And it's absolutely critical to know the ratio of C14 to C12 in the air at the time that an organism was alive in order to date its remains accurately. Now, if we knew how that ratio had changed across the past 50,000 years, if we had evidence of how that ratio had changed over time, then we'd be okay. We could simply make a calibration curve and use that in our calculations. Can we reliably calculate historic atmospheric C14 levels? So, how can we reconstruct something like the historical ratios of C14 to C12 in atmospheric carbon dioxide when we can't go back in time and measure it directly? Well, I suppose one thing we could do is to get a collection of objects whose ages are known independently of radiocarbon dating. Things that can be firmly placed at 1,000 years old, 2,000 years old, 3,000 years old, and so on, all the way back tens of thousands of years. Then, we could measure the C14 to C12 ratio in those objects and combine it with our knowledge of the half-life of C14. That would allow us to independently calculate what the atmospheric C14 to C12 ratio was at the time those objects were alive. We can then use those data to make a calibration curve. So, do these kinds of objects exist? Well, yeah. Human history itself gives us some anchor points. Recorded human history goes back about 5,000 years, and we have artifacts like cloth from Egyptian mummies, Shang Dynasty textiles, or papyrus scrolls that can be historically dated with a high degree of confidence. These provide us with reliable anchor points for what the C14 to C12 ratio was across the span of human history. Beyond that, we have to rely on something called natural clocks. For example, dendrochronology, aka the science of tree rings. Most folks are familiar with tree rings. Every growing season, a tree lays down a new layer of what's called xylem, and it gives us these ring patterns. You can count the age of a tree by counting the number of rings. But these rings aren't a uniform width from one year to the next. Some years have a long growing season. Other years have a short growing season. And here's the cool thing. If you find a slice of wood that's been dead for a few decades, you can actually compare it to a living slice of wood, and you can figure out when your piece of wood was chopped down just by lining up the unique sequence and ratios of tree ring widths. In parts of Europe, they've been able to do this, to line up tree rings in ancient pieces of wood to make a continuous, unbroken chronology of wood samples that go back nearly 14,000 years. So we can test those wood samples all the way across that unbroken lineage of tree rings to get an idea of what the C14 to C12 ratio was across that entire time span. So that gets us back 14,000, maybe 15,000 years. What about before that? There are other natural clocks that can extend this timeline. Corals, for example, build their skeletons in alternating dense and less dense layers, depending on whether it's summer or winter. These density bands can be counted much like tree rings. Because corals take in dissolved carbon dioxide from seawater, and seawater is in constant exchange with the atmosphere, coral skeletons preserve the atmospheric C14 to C12 ratio at the time that they're built. The chemistry is a little bit more complex in water than it is in the air, but the principle is the same. Coral growth bands provide yet another independent record, stretching the calibration data back even further in time. Beyond that, we can use stalagmites as natural clocks. Stalagmites form in caves as mineral-rich groundwater drips from the cave ceiling onto the cave floor. The water contains dissolved calcium carbonate, which in turn contains carbon. During the rainy season, the dripping intensifies, laying down a fresh layer. In dry seasons, the dripping slows or stops, and the layer hardens. Year after year, this cycle repeats, producing a stack of bands like a timeline written in stone. Stalagmites in different caves across the same region will show the same year-to-year banding patterns. That means that we can cross-check them just as we do with tree rings. These stalagmite layers can be counted and tested and give us the C14 to C12 ratio all the way back to around 50,000 years ago. Here's the super cool thing, the super important thing. These dating methods agree when it comes to how the atmospheric C14 to C12 ratio has changed over time. If you go back 10,000 years in tree ring dendrochronology, and 10,000 years in coral reef chronology, and 10,000 years in cave stalagmite chronology, all based on the layers that each process has laid down, and you use each of these to independently derive what the C12 to C14 ratio was 10,000 years ago, they all agree. If we want to go beyond trees, corals, and caves, there are more natural clocks that we could bring into the picture. There are ice cores from ancient glaciers that preserve annual layers of snow and ice that can be counted back tens of thousands of years. Those layers have trapped carbon dioxide in them that can be tested. You also have lake sediments in deep, undisturbed lakes that accumulate yearly deposits as well, rich in carbon dioxide. Each of these provides an additional check, another independent witness to the historical atmospheric C14 levels. So, when you hear creationist claims like,
how much of each of these isotopes was there in the beginning when this creature died? No one knows, because you weren't there to measure it back then.
I wasn't there, you weren't there, but the trees were there to measure it, as were the stalagmites, and the coral reefs, and the lake sediments, and the ice cores. Absolutely, the initial starting condition does need to be considered, and it seems that it is. Are there times when radiocarbon dating doesn't work? All of that sounds very convincing, but what about those times when radiocarbon dating just doesn't seem to work? For example,
we had diamonds, which are supposed to be billion years old, and there should be no carbon-14 in it, and they were analysed, and there was measurable carbon-14 in it. There's good evidence that they're not billions of years old.
We find C14 specifically in samples all throughout the so-called geologic column rock strata, whether it's coal, whether it's diamonds. Diamonds, they're claimed to be billions of years old.
First things first, is this true? Well, perhaps surprisingly, yes. When you try to date very old materials, you can sometimes detect trace amounts of carbon-14. And sometimes when you try to determine the age of contemporary materials, things that should come back as essentially 0 years old, the tests can return ages of several thousand years. So, what's going on here? If radiocarbon dating is supposed to be so reliable, then how could we see these kinds of results? Well, there was a study done by Brown and Sothen at the University of California that does a great job illustrating how and why things like this happen. They wanted to answer the question of contamination. Brown and Sothen radiocarbon dated 4 different materials. First, they used oxalic acid crystals. These are modern, about 60 years old, and serve as calibration standards. Their C14 to C12 ratio should be exactly what you'd expect for something that's essentially new. You could think of them like sugar crystals, but manufactured under controlled conditions. Next, they used sugar crystals, well, table sugar. This came from sugar cane that you figure was harvested probably within the past decade. Like oxalic acid, it should give a radiocarbon age of close to 0. Third, they tested ancient wood. It was a wood sample known to be about 6,000 years old, and that age had already been nailed down through other tools like dendrochronology. It provided a strong benchmark to see if the radiocarbon dating matched the independently known age. Finally, they turned to what's called stone-dead carbon. That would be a material that's far too old to contain any C14 at all. This often includes materials like very ancient coal that was formed millions of years ago, or even diamonds. By definition, these should come back as radiocarbon dead with no C14 in them at all. Here's the twist. They didn't just test these substances once. They tested tiny samples of each, and they also tested large samples of each. That way they could see whether sample size made a difference in the results. Here are their key findings. First, when they radiocarbon dated tiny amounts of new material, like small samples of sugar or oxalic acid crystals, the results came back as much older than the materials really were. Why? Well, they traced the problem to microscopic residues of carbon-12 inside the reagents and the equipment that they were using in the testing process. When you only have a tiny sample, even a little bit of extra carbon-12 contamination will skew the ratio of carbon-12 to carbon-14 in favor of carbon-12, making something modern look artificially old. When they used larger amounts of sugar or oxalic acid, the effect nearly disappeared. The ratios tell the story. If you've got billions upon billions of carbon atoms in a large sample, then adding a few extra thousand carbon-12 atoms hardly matters. The impact of the contamination is drowned out by the sheer size of the sample. Still, the effect leaves behind kind of a background signal or background noise that we can then take into account once we know it's there. Think of it like stepping on a digital scale. Before you weigh yourself, you press the tare button to zero out the scale so that you can get an accurate reading. Next, they found the reverse problem when dating old materials. When they tested small samples of stone-dead carbon, results came back as if the materials were only 30,000 to 50,000 years old. Well, how could that be? Remember, C14 is everywhere. It's like a fine amount of dust floating in the air. In fact, C14 is present in dust. Dust is often generated from dead human skin, or from the excrement of microscopic dust mites, or pollen, or aerosolized dirt particles. Regardless of how much you try to keep the work area clean, and your samples clean, there's going to be some C14 in that sample. Just like how dust settles on a surface. How do we know that's the case? Well, they found that this problem gets much worse when they used small amounts of the test materials. I mean, just a few 100,000 C14 carbon atoms from aerosolized dust can really mess things up, because we're talking about ratios. Again, with larger samples, the contamination effect became much less of a problem. Knowing this allows us to calibrate the instruments to take the background noise into account. The takeaway from Brown and Southen's work is that when you understand and control for these contamination issues, radiocarbon dating works pretty well. Their 6,000-year-old wood sample, for example, dated to the correct age once the tests were run with proper sample size and background contamination eliminated. And remember, radiocarbon dating can be tested and validated at any time. And it is. We have things where we know what the age is, independent of radiocarbon dating, and we carbon date it anyways, and radiocarbon dating agrees, over and over and over. Are there other examples where radiocarbon dating doesn't work? So what about things like this? A freshly killed seal was carbon dated as having died 1,300 years ago. Or, shells from living snails were carbon dated as being 27,000 years old. So does this calibration problem explain our 27,000-year-old snail or the 1,300-year-old freshly killed seal? Well, in those situations, there's something different going on. But the core idea still applies. We need to consider where the carbon is coming from. Take snails, for example. Aquatic snails feed on algae, and biofilms that obtain their mineral nutrients from the rocks that they grow on. Those rocks contain no carbon-14. So when a snail builds its shell using that carbon, it's loading up on extra carbon-12, which throws off the C14 to C12 ratio. It makes it look like the snail is tens of thousands of years old, when it's not. The snail has unintentionally tampered with the ratio because it's fed on radioactively dead, or stone dead, carbon. Seals will show the same effect, but through a different pathway. Seals eat fish, and fish get their carbon from the ocean. Ocean water contains a mix of young carbon that's rich in C14 and very old carbon that's lost all of its C14. This old carbon comes from deep water and from water that's high in dissolved carbonates. This phenomenon has been well documented in marine biology. It's called the marine reservoir effect. So, because the carbon in the ocean is older than what's in the atmosphere, marine organisms, and anything that eats them, can appear hundreds or even thousands of years older than they really are, if the marine reservoir effect isn't taken into account. Incidentally, this comes into play when dating corals, like we talked about earlier. There's just an extra calibration step that's needed. The takeaway. So when it comes down to it, radiocarbon dating is a really well-developed, tried and tested method that uses independent data sources for validation and verification. It accounts for environmental contaminants, both in the environments where objects are found and in the lab where objects are processed. Of course, from time to time, you may hear even stranger and more far-out objections to radiocarbon dating, like the idea that the half-life of carbon-14 has changed over time, or that carbon-14 could turn into other elements besides nitrogen-14, or that objects are prone to acquiring large amounts of additional carbon-14 after they've died. I mean, I suppose folks could really make up any claim that they like, but unless the claim is based in sound reasoning and has evidence to back it up, it should probably be taken with a pinch of salt. That about wraps things up for now, and I hope this has helped in some small way dispel some of the confusion around radiocarbon dating. But if there are any lingering doubts that I didn't cover, or if there are other creationist claims that you find particularly puzzling that you'd like to see covered in a future video, let me know in the comments below. Thanks for tuning in. I'll see you next time.